Hello everyone, welcome to Ibdp Biology. Today our topic is going to be membrane transport part 2. So, in the previous lesson we learned about diffusion, osmosis, and the cytosis and exocytosis. Today we're going to learn facilitated and active transport. First of all, let's start with equilibrium, revise it. Equilibrium is reached when there's an equal amount of particles moving across the membrane in both directions. So in diffusion, during diffusion process, the solute particles move to the higher concentration of the water, and higher concentration of water goes to the lower concentration of water, and when at the end the equilibrium actually reaches. So passive transport is the diffusion of substances across a biological membrane. This occurs without the use of the cellular energy. Passive transport is a diffusion of particles across a biological membrane. It happens without use of cellular energy. There are three types of passive transport: diffusion, osmosis, and facilitated diffusion. We learned the diffusion and osmosis. Today we're going to learn the facilitated diffusion. Facilitated diffusion is one of the four methods of moving particles across membranes. Ions and other particles that cannot diffuse between phospholipids can pass into or out of the cells if there are channels for them through the plasma membrane. These channels are holes with a very narrow diameter. The walls of the channels consist of a protein. The diameter and chemical properties of the channels ensure that only one type of particle passes through, for example, sodium ions or potassium ions, but not both. Because these channels have particles to pass through the membrane from a higher concentration to a lower concentration, the process is called facilitated diffusion. Cells can control which types of channels are synthesized and placed in the plasma membrane, and it's a way that they control which substance diffuse and go out. So now let's watch a video about how facilitated diffusion works.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
Okay, and we've got questions related to facilitated diffusion that we're gonna answer in the check. All right. As you see, the facilitated diffusion happens by the help of the carrier proteins, and these proteins recognize a particular molecule and help it to move across the membrane. The direction it moves is dependent on the concentration gradient, but it's always from higher concentration to a lower concentration. So another example of the facilitated diffusion is actually potassium channels, and potassium channels in axons are voltage gated. They enable the facilitated diffusion of potassium out of the axon. So at the one stage during a nerve impulses, there are relatively more positive charges inside and negative charges outside. This voltage change causes potassium channels to open, allowing potassium ions to diffuse out of the axon. Once the voltage conditions change the channel repeatedly, it closes again. Take a guess about which of the following can diffuse through the cell membrane and the reason. Oxygen, water, glucose, carbon dioxide, sodium, or proteins. So, water, oxygen, it can diffuse small and non-polar. Then we have the water. It can diffuse small pores of an open for it, otherwise it needs help. So the glucose doesn't pass, it's too large. Carbon dioxide can diffuse small and non-polar. Sodium cannot pass its charge, and glucose cannot pass, it's too large. Active transport, the movement of a substance across a biological membrane against its concentration or electrochemical gradient with the help of energy input and specific transport proteins. This movement of particles occurs against the concentration gradient or from an area of lower concentration to an area of higher concentration with the use of ATP. An adenosine triphosphate. Adenine containing nucleoside triphosphate that releases free energy when its phosphate bonds and hydrolyzed. This energy is used to drive endergonic reactions in the cell. The primary active transport requires ATP. Integral protein pumps use the energy from the hydrolysis of ATP to move ions or large molecules across the cell membrane. Molecules are moved against their concentration gradients. Now, let's see the video about the active transport. Let's see how the primary active transport starts. So this is the intercellular fluid and extracellular fluid, and there is a protein channel, and you see what happens. The ATP binds to a protein channel. By hydrolysis it becomes the ADP. Then let's see how it happens. So then the ion is actually bind to it and it opens. It goes into the potassium, goes inside. Then another ATP hydrolysis happens. Another potassium ions goes inside. This is actually happens like that, and these are actually questions that you're going to answer.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Alright, that's how we see the sodium-potassium pumps actually work in active transport, and there are the questions that we're going to answer. So this is the structure and function of the sodium-potassium pumps for the active transport and potassium channel for the facilitated diffusion in axons that we saw in the animation. The cells can hydrolyze ATP, so it means it is break down ATP and use the energy released to move substances across the plasma membrane against the concentration gradient. We know that the normal concentration gradient is when the molecules move from the higher concentration to a lower concentration, but here it's going to happen from the lower concentration to a higher concentration, and the energy is used to change the shape of the membrane protein is a pump. Each pump only transports particular substances. The particle will enter the pump on the side with a lower concentration and bind to a specific site for that type of a particle. Energy from ATP is used to change the shape of the pump. The particle is released on the side of the higher concentration and the pump returns to its original shape. An example of this is the sodium-potassium pump in animal cells. There is a higher concentration of potassium ions inside the cell and a higher concentration of sodium ions outside the cells. And what happens? The cell maintains this concentration using the sodium-potassium pump membrane protein. So what happens during the sodium potassium pump? The sodium ions are going outside for three sodium ions, 2 potassium ions going in. For 3 sodium ions going outside the cell, 2 potassium ions get inside the cell. So they are working against their gradient. So this is how actually it happens. So the cytoplasmic sodium binds to the sodium-potassium pump. The sodium binding stimulates phosphorylation by ATP. So the ATP is actually broken down to ADP. So potassium is released and sodium side are receptive again. The cycle repeats. Phosphorylation causes the protein to change its conformation and expelling sodium to the outside. Loss of the phosphate restores the protein's original conformation. Extracellular potassium binds to the protein, triggering release of the phosphate group. So this is how actually the primary active transport happens, but now I'm going to show you the secondary active transport by animation. So here we see the lower intercellular sodium concentration and high extracellular sodium concentration, the intracellular fluid and extracellular fluid, and how it happens. Let's see. When sodium is like getting inside the intracellular fluid. From high extracellular sodium concentration to a lower intracellular sodium concentration. So there are the questions actually that you're going to answer as well, as you see here. So guys, so we watched all the animation and videos. Thanks for watching, guys. Have a nice day. Bye bye.